Glutamate is the principal excitatory neurotransmitter in the mammalian central nervous system, and its release from neurons is a tightly regulated process essential for synaptic transmission, plasticity, and overall brain function. The mechanisms governing glutamate release are intricate, involving both classical and non-classical pathways. The primary mechanism of glutamate release is calcium-dependent exocytosis at presynaptic terminals. Upon arrival of an action potential, voltage-gated sodium channels depolarize the neuronal membrane, leading to the opening of voltage-gated calcium channels, mainly PQ-type and N-type, at the active zones. The influx of Ca2+ into the presynaptic terminal triggers a cascade wherein synaptic vesicles, loaded with glutamate via vesicular glutamate transporters, VGLUTs, are mobilized toward the plasma membrane. The SNARE protein complex, comprising synaptobrevin on the vesicle, syntaxin, and SNAP25 on the plasma membrane, mediates vesicle docking and fusion. Synaptotagmin, acting as a Ca2+ sensor, facilitates rapid vesicle fusion upon calcium binding, resulting in the exocytosis of glutamate into the synaptic cleft. Beyond this classical pathway, several modulatory processes influence glutamate release. Presynaptic receptors, such as metabotropic glutamate receptors, MGLURs, GABA-B receptors, and various neuromodulator receptors, adenosine and endocannabinoid receptors, can inhibit or enhance glutamate release via second messenger systems that regulate calcium channel activity or directly modulate the SNARE machinery. Additionally, phosphorylation of presynaptic proteins by kinases, such as pKa and pKc, can alter release probability. Under certain conditions, non-vesicular release mechanisms also contribute. For example, reversal of glutamate transporters, notably under pathological conditions like ischemia, can lead to glutamate efflux. Astrocytes and other glial cells may release glutamate through hemichannels, volume-regulated anion channels, or even exocytosis, though these mechanisms are less prominent under physiological conditions. Altogether, the release of glutamate from neurons is a highly orchestrated process, integrating electrical activity, intracellular signaling, and feedback from the synaptic environment to ensure precise control over excitatory neurotransmission. Dysregulation of these mechanisms can result in excitotoxicity and contribute to neurological disorders such as epilepsy, stroke, and neurodegenerative diseases.